Hello guys and welcome back to DNA replication, and today we are going to do the lecture 8 in this. Okay, so I need you to brush up like what all we have discussed till now. So we have discussed about the origin in the case of prokaryotes, in the case of eukaryotes, right? We have also discussed about the replication for different DNA polymerase, everything, right? Now in this we are going to talk about the eukaryotic. Okay, till now we have discussed about the prokaryotes. Now we are going to discuss about the eukaryotes, origin and initiation. So already we have discussed. So in prokaryotes, so how many origin of replications are there? How many origins are there?
One.
Very good. And what about in eukaryotes? What is the number?
Multiple.
Yes, it is multiple. There is no definite number for this. Okay, so you have got multiple origin of replication. So in eukaryotes, the clusters of about 20 to 50 replicons initiate simultaneously at defined times throughout the S phase. So it says that you have got around 20 to 50 replicons. What are replicons? Replicons are the site at which the origin will be recognized, the sequence will be recognized, and all the replication associated proteins will come and bind. Okay, so that is your replicon. And always, please always remember that in the S phase, your DNA replication is happening. Okay, now why it is important to remember? Because once we start doing cell bio, in that we have got cell cycle, right? So when we are going to talk about cell cycle, then you need to merge the information. So right now I just tell you that cell cycle is divided into two phases, interphase and the M phase. In the interphase, you have got the G1 phase, then S phase, G2 phase, and then comes your M phase. Okay, then again back to G1 for the new round of replication by the daughter cells. Okay, so this entire G1 to G2 phase is known as the interphase. There, we need to remember the proteins which are involved in the DNA replication, because it's not that single protein, they are going to function only one particular thing. Okay, they might have multiple roles also. जैसे कि एक protein के पास दो तीन काम होंगे. So in order to help in the DNA replication, it is also helping in DNA repair, or maybe it is helping in the recruitment of the other proteins. So ऐसा हो सकता है. Okay. So those which replicate early in S phase comprise predominantly euchromatin, which includes transcriptionally active DNA, while those activated late in S phase are mainly within the heterochromatin. Now, who can tell me what is euchromatin and what is heterochromatin? Yes, anyone? Yes, Tanvi? Euchromatin region in DNA or heterochromatin region in DNA क्या होता है? See, it is important because next what we are going to learn after this DNA replication, it is going to make a huge difference. So it's very basic and everyone should know about it. So Tanvi, what is it? Anyone? Akshay, Shruti?
Ma'am, the euchromatin is the transcriptionally active one, which means that it is not as constricted by the histones and actually builds proteins, while heterochromatin is very condensed and methylated and is it doesn't produce proteins. It is there, but it doesn't transcribe anything.
So don't say produce, it doesn't encode. Okay, so DNA is going to encode. Yeah. The terminology is important. So very good, Shruti. So euchromatin regions are those which are transcriptionally active, right? Now they are saying that those which replicate early in S phase comprise predominantly euchromatin region. So of course they are going to be transcriptionally active. Now what kind of genes are going to be active at the early of S phase? And like already it is given, centromeric and telomeric. So centromere and telomeres are the region which comes under the heterochromatin. So they are going to replicate at the last. So the centromere region as well as the telomere region, they are going to replicate at the last. Okay. Now the question is, which kind of genes or that are going to be transcriptionally active DNA? Like what kind of genes can you expect to be replicated or to be encoded before the DNA synthesis starts? Any guesses? If I have confused you guys, question is, what kind of genes are coming into this euchromatic region, okay, which is happening or replicating early in the S phase, and why? ऐसे कौन से जीन्स होंगे जो डीएनए रेप्लिकेशन से पहले एनकोड होने चाहिए या एक्टिव होने चाहिए? नाउ द क्वेश्चन इज क्लियर?
The ones required for DNA replication.
Exactly, very simple. Yes. So what kind of genes which are responsible for the DNA replication? So you need to know what are the proteins required for DNA replication. Okay. So like DNA poly हो गया आपका, different types of DNA polymerase हो गया. So like in the case of eukaryotes, you have got alpha, beta, gamma, delta, epsilon. These are the DNA polymerase. Okay. There are 5 dominant DNA polymerase in the case of eukaryotes. Then you have got the ligase, you have got the topoisomerase, etc., etc. So all these need to be present before your DNA replication starts. Okay. So इसी वजह से those regions are coming under the euchromatic region. Now is it clear? See, I was stressing this sentence because this we have asked during the interview once. That's why. Because we are saying that the DNA is undergoing replication. After replication only those regions are free for the transcription, right? So पहले से ही DNA polymerase कहाँ से आता है? पहले से ही आपका topoisomerase ligase कहाँ से आता है? So that was a question we asked during the interview. Okay, I hope you got the concept over here. Is it clear to everyone?
Yes, ma'am.
Yes, ma'am.
Okay.
Yes, ma'am.
So individual yeast replication origins have been cloned into the prokaryotic plasmid, and they came to know that they are known as autonomously replicating sequences. Okay, so इसमें क्या होता है? So it is difficult to grow yeast and to grow and culture mammalian cells. So सबसे आसान तरीका क्या होता है? So put that sequence, okay, just the sequence into the plasmids of the bacteria, because they can divide faster and you can get the result fast, right? So this is how we came to know about the proteins which are responsible in the yeast and in the eukaryotic mammalian cells, like DNA replication में कौन-कौन से प्रोटीन लगते हैं, ठीक है? तो ये बेसिक है. मोल बायो में कोई भी सीक्वेंस, कोई भी जीन के बारे में अगर आपको स्टडी करना है, whether it is from a mammal, whether it is from a plant, a yeast, any species, okay, eukaryotic species, what we do, we get the sequence, we get the gene into the plasmid, and this plasmid will put into the bacteria so that we can do the study faster. Otherwise, yeast को grow करना it's difficult. Mammalian cell को grow करना difficult because there are the chances of contamination is so high and it takes time, right? 24 घंटे लगते हैं from one cell to become a 2 cell. Whereas in the case of bacteria, just imagine how fast it is, right? In a hour, 3 rounds of replication or 3 rounds of generation will be over in the case of bacteria. So इसलिए सारे studies जो होते हैं वो हम बैक्टीरिया में करते हैं. Okay, so in the case of yeast, जैसे कि बैक्टीरिया में it was ORIC, right? Remember ORIC, the starts of the origin of replication in the case of bacteria. In the yeast, it is known as autonomously replicating sequences, and the minimum length of DNA that will support replication is around only 11 base pair and has got this sequence. Okay, याद करने की जरूरत नहीं, just for the reference, you don't have to by heart the sequence. Okay, so now the sequence is bound by the origin recognition complex. कौन सा? जो आपका जो हमने अभी शुरू किया है, ARS. Okay, origin recognition complex. So these are the proteins which will come slowly and will bind to the entire ARS. Okay, and the entire thing will finally be known as replisome. Okay, we'll come to that. So the sequence is bound by the origin recognition complex, which when activated by CDK. Now, what is CDK? Can anyone tell?
Cyclin dependent kinase.
Very good. So it's a kinase, right? So it is going to start or permits the opening of the DNA for the copying. So what does it mean? It permits the opening of the DNA. It is activating your helicase. Okay. So defined origin sequences have not been isolated from mammalian cells, and it is believed that initiation of each replicon may occur at random. And this is why it was difficult for scientists to get the exact sequence of the ARS or ORC. ठीक है, अभी जैसे कि in the case of bacteria we can say that ये ORC है, ये termination sequences है, but in the case of mammalian DNA it is difficult to say, क्योंकि if suppose the replication is starting this time over here, next time it will be starting somewhere else. So it is a kind of random process. It is not like, you know, fixed, it is random. So in contrast to prokaryotes, eukaryotic replicons can only initiate once per cycle. So इसके केस में जो हमने पढ़ा था, like in contrast to prokaryotes, do you remember, like once the replication started, जैसे ही आपका फर्स्ट खत्म होने वाला था तब तक आपका दूसरा भी शुरू हो जाता है, right? In the case of E. coli, I have explained like why the generation time is 40 minutes, but we see that the E. coli divides in 20 minutes. Remember the diagram?
Yes, ma'am.
हाँ. So in contrast to that, we can say that eukaryotic replicons or eukaryotic DNA is going to replicate only once per cycle. ठीक है? And to ensure that, we have got around millions of proteins to ensure this thing. Okay, we'll talk about few of them. Okay, so protein, which is also known as the licensing factor, is absolutely required for initiating and inactivating after use. Only gain access to the nucleus when the nuclear envelope dissolves at mitosis, thus preventing the premature reinitiation. So what does it mean? See guys, if you remember in the case of eukaryotic cell, your DNA synthesis is happening in the S phase, right? So after the checkpoint in the G2 phase, once it is done, it decides, or it is, you know, allowed to go into the M phase. Now, what is M phase actually? M phase is the mitosis. And we know that in the case of mitosis, the nuclear envelope is going to get disappear, right? Right? So it is a protein which is required and it's inactivated. Otherwise, what will happen? This again protein will go and initiate the replication. अब इसमें क्या होता है? Nuclear envelope का disappear हो गया. Why? Because it is going to get condensed and form into a chromosome. पहले आपका क्या है? It is a chromatin structure, right? So slowly it gets condensed into a chromosome, and then only the mitosis will happen. यहाँ पे आपका separation will happen for the daughter cells, right? So nuclear envelope disappearance or the lamins, basically we are going to talk about lamins. They get inactivated, okay? So they are going to make sure that the premature reinitiation will get prevented. Are you getting the point over here? Do you know what are lamins? Yes, Akshaya, lamins क्या है?
I don't know, ma'am.
Okay, anyone? Tanvi?
No, ma'am, I don't know.
Okay, no problem. So, okay, do you remember that, okay, do you know that in the case of eukaryotic cell, you have got the nucleus, and this nucleus is made up of nuclear envelope? ये हमें मालूम है, right? Now, this nuclear envelope is nothing but made up of, इसके नीचे, see, you all know that the cell is made up of plasma membrane, right? So mitochondria has got double layer of plasma membrane, chloroplast has got double inner and outer plasma membrane, right? In the same way, nucleus is also having a double, right? Now, below this envelope, okay, below this envelope, there are proteins called lamins. Okay, now imagine have you guys seen like how a building is being constructed or how any house is being constructed? कभी देखा है? So what do you see? ऊपर की छत बनाने के लिए they keep the bamboos like this over here, and over here also they keep to have that ceiling. तो यहाँ पे bamboos रहेंगे ऐसे interconnected. Okay, if suppose this is your building and this is your ceiling, okay. So they are going to keep the bamboos over here for the support and over here for the support. Have you seen the construction of any building or any house? आपको idea आ रहा है मैं क्या बताना चाहती हूँ?
Yes, ma'am.
हाँ, तो ऊपर की छत बनाने के लिए you need to have the support, right? and then to इसको support करने के लिए नीचे हम लोग bamboos लगाते हैं. तो same चीज, if I don't have lamins, okay, if I don't have lamins, what will happen to the nucleus? It's a 3D structure. If I don't have lamins all over around the nucleus, what will happen? This will become flat. वो एक दूसरे में चिपक जाएगा. Okay, to keep it into a spherical shape or whatever the shape the nucleus is having. Okay, to keep it in that intact shape, I have got lamins over here. ऐसे. ठीक है? And again, they have got certain other proteins which is holding these lamins ऐसे ऐसे. अब इस बीच में you have got your chromosome, sorry, chromatin or DNA, which later becomes chromosome. Okay, so this much is clear. What are lamins? We'll come to that lamins. So lamins are actually your intermediate filaments. In cell biology, we'll talk about microfilaments or actin filaments. Suna hoga aapne actin filaments. Then we have got the intermediate filaments. For example, keratin ho gaya, जो आपके nails and hair में present है. So similarly, we have got lamins. Okay, and then we have got the microtubules. जिससे आपका what do you say, the spindle fibers are formed, right? In few cases, it is also helping in the flagellar movement or the motor movement. Okay, so lamins are your intermediate filaments. So we'll come to that once we start doing the cell biology. Abhi itna basic clear hai that below the nuclear envelope, you have got the lamins, which is helping in maintaining the structure of the nucleus. Yes?
Yes, ma'am.
Okay, so there are many proteins which make sure that these lamins are then phosphorylated. Okay, now once they are phosphorylated, of course, they are going to be the kinases. Okay, हम धीरे धीरे जाएंगे, like which kinase is phosphorylating what. So we have got lamins A, B, and C. Okay, we'll talk about the structure of lamins later. So we have got three types of lamins. A and C are together. इनको support करने के लिए B रहता है. Okay. Now once these A, C, and B they get phosphorylated with the help of the kinase, what will happen? They start getting ये गिरने लगते हैं. They start falling apart. So what will happen? Then is your nuclear envelope will start getting disappear. ठीक है? So disappear होना मतलब क्या होता है? They are getting fragmented. They are not like exactly ऐसे गायब नहीं होते हैं. They just start getting fragmented. अब वो छोटे छोटे phospholipids and छोटे छोटे protein में आ जाते हैं. Okay. Now once the chromosomes come together after the separation, then again they come back together. So there is a beautiful animation also. I'll show that later in the cell bio section. Okay. So there are proteins which will phosphorylate this and make sure that the DNA replication or the initiation is not happening. Okay. Is this much clear?
Yes, ma'am.
Yeah. Okay. Now let's talk about the replication fork. Replication fork. So, what all you remember about the replication fork about the prokaryotes? क्या याद है? What all is present in the replication fork of prokaryotes? Anyone? Like what kind of sequence is present? सबसे पहले कौन सा protein आता है? What happens? Shruti, can you tell?
Yes, ma'am. So first, there's the 5 9-mer sequences where DNA A binds over there, and then there's the 3 13-mer sequences where the DU sequences are.
Yes. Then? So DNA A binding protein will start binding over here. They'll start forming a loop-like structure. Then what will happen? Then after DNA A, DNA C is there. which recruits your B, right?
Yeah.
Yeah.
And
So
DNA B then, yeah, it's the one that unwinds the DNA. And then primase is here, the DNA G, which RNA primers are created by primase.
Yes.
And then to keep everything from ligating together, SSB proteins are also there.
Yes.
I think, yeah, and then DNA gyrase is there, which as the DNA is formed and the tension keeps coming up because it's circular DNA, so that stress is relieved by DNA gyrase. And then the DAM methylase is methylate the daughter strand so that
yeah. So when you say about the tension in the DNA, the best word is to use supercoiling. Okay.
Yeah. Okay.
Good. So now let's talk about, like, in the case of the eukaryotes. So before copying the DNA, the DNA must be unwound from the nucleosomes at the replication fork. So yes, in the case of eukaryotes, we have got the nucleosomes. Okay. It is very beautifully given in the Watson. It's a chapter 8, I guess. So nucleosomes, you should know that there are 4 types of proteins are there. Okay. And what are the basic features about these proteins? That they are positively or positively charged amino acids. Okay. So what are the positively charged amino acids? What are the examples?
Arginine, lysine,
and arginine.
And
histidine.
Yes, very good. Histidine, right? Okay. So in the case of the nucleosomes, they are basically rich in arginine and lysine, and nucleosomes, they have got the 4 types of the protein. So what are those? H1, H2A, H2B, H3, and H4. Okay. So among these 4 are going to form the nucleosomes. So of course, the H2A and H2B forms the heterodimer, and H3 and H4 forms the heterodimer, whereas H1 is acting as the linker between 2 nucleosomes. Guys, you have got the idea, right, about the nucleosomes?
Yes, ma'am.
आपको पता है ना? Okay, good. So, so you know that H1 is acting as a linker, H2A, H2B, and H3, H4. But we know that in the case of the nucleosomes, it is अभी ये 2 protein हो गए, ये 2 protein हो गए. So what does it mean? It's a tetramer, right? But you should always remember that nucleosome is actually a octamer. So what does it mean? कुछ गलत हो गया क्या? Now if I say that nucleosome it's an octamer, so octamer मतलब कौन से प्रोटीन कितने होंगे फिर? Yes.
All of them will be 2 in number, ma'am.
Yes. So कैसे होता है? Like this is your H2A, H2B. यही आपका ऊपर नीचे रहेगा, ठीक है? Then you have got along with this H3H4, H3H4. So like this, you are going to have the octamer, and your DNA is going to bound like this. तो ये एक न्यूक्लियोसोम हो गया, ऐसे ही दूसरा होगा, और इन दोनों के बीच में you are going to have the H1 linker. इतना क्लियर है? I am not that good in drawing, but I'm sure you must have seen, right? Now, before I proceed, there is one more thing which is important for me to discuss over here. So now you know nucleosomes and around how much base pair the DNA is wrapping. That is around 147 base pair. ठीक है? अभी इस पे बेस क्या है ना, बहुत सारे न्यूमेरिकल्स आते हैं, तो एक डेमो में दे देती हूँ, and the rest you can practice from that playlist is there, right, where I have mentioned about the numericals from biochemistry and mole bio. So please practice from there. Many questions have been uploaded. So one question I'll give you is that, so there are around 200 H3 proteins, okay? How many nucleosomes will be present? ये बहुत ही simple basic question है. Sorry. Yeah. सोचो.
I'm 100.
Sure.
Yes, ma'am, because each nucleosome contains 2 H3 proteins and we have 200.
Will be half.
Yes. So आपके पास कितना हो गया? तो आपके पास हो गया आपके पास around 100 nucleosomes हो गए. ठीक है? Good. Now, if I give you, for example, like दूसरा question में देती हूँ. So it was simple, but it took time for you to calculate, right? थोड़ा ध्यान देने में, but it's good. Okay, तो इतना simple तो कोई नहीं पूछेगा आपको, obviously. Now, एक क्वेश्चन था जो हम लोग ऐसे बेसिकली ऐसे इंटरव्यू में पूछते रहते हैं, that how many nucleosomes are present in a mammal cell? कैसे कैलकुलेट करोगे? हाँ, अब शुरू कहाँ से करोगे? तो आपके इजी कैलकुलेशन को let us say that, let us assume that one nucleosome 200 बेस पेयर है, ठीक है? अभी 147 रहता है तो कैलकुलेशन में डिफिकल्ट होता है, बट 200 बेस पेयर बी कंसीडर्ड बिकम्स इजी. नाउ व्हाट शुड बी द नेक्स्ट स्टेप? यस, ओके. एक काम करते हैं, आपके लिए मैं असाइनमेंट रखती हूँ, एंड देन टुमारो बिफोर स्टार्टिंग वी कैन स्टार्ट विद दिस. सो योर क्वेश्चन इज हाउ मेनी, please note down, okay, how many nucleosomes are present in a mammalian cell? Okay, consider one nucleosome 200 base pair. तो सब सेकंड स्टेप क्या होना चाहिए आपका यहाँ पे? how many base pair are present in a mammalian cell? Then you should know how to calculate. ये हो गया आपका फर्स्ट क्वेश्चन. सेकंड क्वेश्चन में देती हूँ आपको. so you need to calculate or enumerate the number of nucleosome. Again, कहीं कहीं पे nucleosome को उन्होंने beaded बोला है, ठीक है? beaded structures. So don't get confused. nucleosomes या beaded structure same हो गए. So enumerate number of nucleosome or beaded structure present in a nucleus. Okay, present in a nucleus having base pair of 2.4 into 10 raised to 6 base pair. तो अगर आपको पहला वाला आ जाएगा तो आपको डेफिनेटली सेकंड वाला भी आ जाएगा. इज द क्वेश्चन क्लियर टू ऑल ऑफ यू? यस,
Yes, ma'am.
हाँ, क्वेश्चन क्लियर है ना आपको? दोनों क्वेश्चन सेम है, इट जस्ट दैट द नंबर इज डिफरेंट. ओके, सो जस्ट ट्राई टू गेट द आंसर कि आपको कितना न्यूक्लियोसोम लगेगा जब इतना बेस पेयर दिया हुआ है. ओके, चलो let's go back to our topic now. Okay, okay, so nucleosome, the presence of the nucleosomes in the eukaryotes is going to slow down the movement of the forks to about 50 base pair per second. Okay, now after the fork has passed, New nucleosomes are assembled from a mixture of old and newly synthesized histones. This is also really very important because कैसा होता है that on the parental strand and on the daughter strand. So nucleosomes क्या है? They are just like the histone proteins are separated. ठीक है, आपका DNA straight हो गया so that the replication can start. Now once the replication is done, what happens? You have got the parental strand and the daughter strand, right? यहाँ पे and यहाँ पे आपका daughter होगा, यहाँ पे एक parental होगा. Okay. So total ऐसे हमें मिलेगा. Now the thing is that because there is a memory in the parental strand, there is a memory which is present in the parental strand. Now when I say memory present in the parental strand, what does it mean? It means it is a memory sequence which is present in the parental strand which will recruit back the histones. ठीक है, जिसकी वजह से your this DNA and this DNA is going to have mixture of new and old histone proteins. Is it clear this much? Okay. Should I explain again?
Yes, ma'am.
हाँ, मतलब explain करना
Yes,
है, right?
ma'am.
Yes, ma'am.
Okay. I'll take the whiteboard. Okay. See. Whatever I'm telling right now is just the gist of the story. Okay, detail में इसको करना बाकी है. Okay, so we know that आपका replication हुआ यहाँ पे. So finally, this is going to be your parental strand. एक यहाँ पे आपका parental strand होगा, right? इसके ऊपर आपका daughter strand बना. Here also daughter strand बना. Correct? Let us say that okay, this is your parental. And जो रेड वाला है वो आपका डॉटर स्ट्रैंड हो गया. ओके, नाउ आपका रेप्लिकेशन यहाँ पे आगे आगे चलता जा रहा है. ओके, लेट इट बी देयर. नाउ सो बिफोर इन द केस ऑफ यूकैरियोट्स, बिफोर द रेप्लिकेशन स्टार्ट, द हिस्टोन्स जो रहेंगे उसको आपने हटा दिया, राइट? दे आर प्रेजेंट इन द न्यूक्लियोप्लाज्म. इतना एग्री कर रहे हो? Then only you are able to get a straight DNA for the replication.
Yes, ma'am.
Right? Now, your replication fork has going आगे जा रहा है. अभी पीछे क्या होना है? अभी पीछे जैसे जैसे आपका DNA replicate हो रहा है, it needs to get condensed. Okay? In the case of eukaryotes, it is possible with the help of the histone proteins. Okay? Now, histone proteins यहाँ पे कैसे है? See, your cell understands that DNA is undergoing synthesis. Right, replication. So of course मुझे और histone proteins लगेगा. So that will be transcribed and translated, and those proteins will be ready. आपके histones जो ready होंगे, ठीक है? Now यहाँ पे क्वेश्चन आना चाहिए, histones जो proteins है वो तो translate होते हैं, वो तो cytoplasm में रहते हैं, right? But as your cells is undergoing synthesis, the recruitment of the protein, which is also known as the import of the protein, okay, is happening. So आपके जो प्रोटीन्स जो ट्रांसलेट होके साइटोप्लाज्म में है उसको हम इंपोर्ट कर लेते हैं न्यूक्लियोप्लाज्म में. ओके, तो उसका कोई प्रॉब्लम नहीं है. नाउ जो पैरेंटल स्ट्रैंड्स में जो आपके न्यूक्लियोसोम्स है, लेट अस से दैट दे आर ऑफ दिस कलर. ओके, नाउ व्हाट विल हैपन? जस्ट इमेजिन दैट दिस इज अ ऑक्टामर. ओके, दिस इज ऊपर 4 नीचे 4 ऐसे इमेजिन कर लो. नाउ व्हाट विल हैपन? Once they get dissociated, they will come back. और ये कैसे होता है? Because of the memory sequence which is present on the parental strand. ठीक है, अब क्या होगा? Since you have got the parental strand and daughter strand, the chances of having new and old histones randomly is possible. तो अभी क्या होगा? The chances ये है कि एक मेरे पास old आएगा और बाकी मेरे पास new आएंगे. तो यहाँ पे फिर कैसे होगा? तो चांसेस है कि यहाँ पे एक आएगा और यहाँ पे मेरे पास ओल्ड आएंगे हिस्टोन्स, एंड देन दे विल रैप अराउंड ऐसे. अब भी क्लियर हुआ?
Yes, ma'am.
कहाँ पे डाउट आ रहा है? लाइक यू कैन टेल मी दैट आल्सो, लाइक व्हिच पार्ट इज नॉट क्लियर. यस यस तन्वी, श्रुति, अक्षय, पूरा क्लियर है या कोई पार्ट जो है जो नहीं क्लियर हुआ है? So clear है to everyone? Okay.
Yes, ma'am.
Oh. Yeah. Okay. Now, I hope you can see the screen, right? You can see the slide.
Yes, ma'am.
Okay. So now over here we have got the replication protein A along with the DNA helicase and single stranded binding proteins. We also need to remember about the RPA, that is replication protein A. And guys, one more request is there. Please try to remember all the short forms or the acronyms in the mole bio. ठीक है, बहुत सारे आएंगे and those are specific for mole bio. अच्छा, okay. Now, and then we have got the 3 different DNA polymerases which is involved in the elongation. Now there's something called polymerase switching. That is also a very important topic which we'll discuss in this upcoming lectures. Okay. Okay. So what are those 3 DNA polymerase? So we have got the alpha, delta, and epsilon. Okay. वहां पे कौन से थे? We had polymerase 1, 2, and 3. Here we have got alpha, delta, and epsilon. So the leading strands and each lagging strand fragment are initiated with RNA by primase activity, which is done by the polymerase alpha. Okay. वहां पे क्या था आपका DNA G, which has got the primase activity. Here it is your DNA polymerase alpha. Okay, just compare and study so that you can remember. Now this polymerase continues elongation with DNA, but it is quickly replaced by delta. Okay, on the leading strand and also on the lagging strand. Now the role of DNA polymerase epsilon is less clear, but it may simply complete the lagging strand fragments after the primer removal. Both these enzymes have associated with the proofreading activity. Okay.
Ma'am,
Huh.
I have a doubt, like how DNA polymerase can add a RNA strand, ma'am? The primer.
DNA polymerase alpha नाम दिया है, but actually it's a protein only, it's an enzyme only, right? जैसे DNA
okay.
G वहां पे प्रोटीन था, it's just that उसका नाम DNA G रखा है, but it is actually basic level वो काम क्या करता है, वो प्राइमेज है, प्राइमर ऐड करता है. यहाँ पे इसका नाम क्या दिया हमने, DNA polymerase alpha नाम दिया है, but वो भी बेसिक लेवल में प्राइमेज ही है ना?
Okay.
हाँ, now it's clear?
Yes, ma'am.
हाँ, actually they are protein, ये याद रखो, they are protein, they are enzymes. Okay. हाँ. Okay. One second. Huh? Okay. So the ability to synthesize long DNA is conferred on the DNA polymerase delta by pCNA. Okay. Proliferating cell nuclear antigen, which is the functional equivalent of the beta subunit of E. coli, DNA polymerase 3 holoenzyme. Yes. कुछ याद है? What is the beta subunit of DNA pol 3 holoenzyme? Right, you need to remember, otherwise difficult होगा. Okay. Do you remember something called like sliding clamp? अगर ये DNA pol था तो उसके नीचे के ऐसा structure हम बनाते थे, which is helping in the, you know, so that it is associated with the DNA and does not fall off from the strand while replicating. कुछ याद आ रहा है?
Yes, ma'am.
हाँ?
Yes.
Okay. And इसके पीछे होता था हमारा clamp loader, which is having the tau protein जो आपको hold करता था. Okay. So here we have got the clamp loader. यहाँ पे hold करने के लिए आपका tau protein होता था, which is holding the sliding clamp and DNA polymerase. So इस तरह से 3 थे यहाँ पे. Okay. So 2 lagging के लिए, 1 leading के लिए. Now just imagine the same thing. जो हमने beta subunit sliding clamp हम बोल रहे हैं, beta clamp बोलते हैं जिसको. It is nothing but pCNA in the case of eukaryotes. ठीक है? Full form भी थोड़ा याद रखना, because ये बीच में आगे पूछा जाता है, and it is also responsible for, like, you know, when we are going to talk about the repair and cancer वगैरह वगैरह. So it is important over there. So it is the proliferating cell nuclear antigen. So the small size of eukaryotic lagging strand fragments appears to reflect the amount of DNA unwound from each nucleosome as a per progress. In addition to doubling the DNA, the histone content of the cell is also getting doubled during the S phase. Obviously, right? तो यही मैंने अभी आपको बताया था कि जैसे जैसे आपका डीएनए डबल हो रहा है वैसे ही सारे प्रोटीन्स विच इज रिक्वायर्ड, क्योंकि अभी आपका एक डबल हेलिक्स से दो डबल हेलिक्स होने जा रहे हैं. सो ऑब्वियसली द प्रोटीन्स विच आर एसोसिएटेड विद DNA is going to get double. Okay. Okay. So these are the names and all. If possible, try to remember, but itna important nahi hai. Jahan bhi important hoga, let you know, guys. Okay. It is just for the the comparison they have given. Like mammary name alpha, beta, gamma, delta, epsilon. जिसमें से मैंने पहले mention कर दिया है that alpha, delta, and epsilon are more important. ओके उसमें से भी आपका डेल्टा काम का है मेनली. जैसे प्रोकैरियोट्स में डीएनए पॉलीमरेज 3 इंपॉर्टेंट था, यहाँ पे डेल्टा इंपॉर्टेंट है. ओके तो दिस इज योर प्रिंसिपल रेप्लिकेटिव पॉलीमरेज. यहाँ पे आप देख सकते हो वो प्राइमिंग में हेल्प करता है एक्सेट्रा एक्सेट्रा. ओके now let's talk about the real eukaryotic replication. Okay. So when we say replosome assembly, we are just talking about how the eukaryotic initiation is happening. Okay, so we have got the ORC. So guys, ORC क्या था? What was ORC?
The origin of replication, where it starts, initiates.
नहीं, ORC you can see the figure. ORC देख के क्या लग रहा है? क्या है वो?
The entire complex from which replication starts.
हाँ, but it is also known as what? Like उसका full form क्या था? Yes, नहीं याद आ रहा. It is the origin recognition complex, right? And it is binding where ARS. याद आ रहा है कुछ? है ना? ARS, autonomous replicating sequences. तो ARS जहां जहां पे present है वहां पे आपका ORC आएगा. ठीक है? Now, once this is happening, you can see in the figure also that it is known as the helicase loading. Okay, so in case of eukaryotes, the helicase loader is MCM27. वहां पे कौन था? प्रोकैरियोट्स में हेलिकेस डीएनए बी था ना?
Yes, ma'am. DNA B.
हाँ, एंड यहाँ पे इट इज नोन एज MCM27. Now, what is MCM27? हाँ, yes.
MCM2-7 लिखा है ना, तो they are multiple, right? 2 to 7 of them.
हाँ, yes. You can see the figure also, na? So many multiple complex are there. 27 is 27 नहीं है. 2 to 7 there are many are there. Okay, yeah. Now, what does MCM stands for? Yes, it is the mini chromosome maintenance. क्या है? मिनी क्रोमोसोम मेंटेनेंस, यानी कि वो क्या कर रहा है? मेंटेनेंस वाला काम कर रहा है. ठीक है. सो अभी कैसा होगा यूकैरियोट्स में? यू विल बी लाइक थिंकिंग ये कैसा नाम है बट काम क्या है? ओके, इट जस्ट बिकॉज जैसे जैसे साइंटिस्ट को जो डिस्कवरी होती गई एंड व्हाट दे फेल्ट लाइक इट इज सिमिलर टू व्हाट एंड जिसके साथ सिमिलैरिटी मिली वैसे वैसा उसका नॉमेनक्लेचर कर दिया है. सो डोंट वरी अबाउट इट. Okay, so it is mini chromosome maintenance proteins and they have got the evolutionary conserved domain. So जो conserved रहते हैं उनका ये नंबर दे देते हैं. Okay, okay, इतना क्लियर है? Yes,
Yes, ma'am.
okay. Now let's talk about next step. अभी हेलिकेस लोड हो गया है. Now it is loaded, but it is not activated. Okay, your helicase is just loaded onto the double-stranded DNA, जहाँ पे उसका replication शुरू होगा, but it is not activated. Now what will happen? The kinase is coming. Okay. One more thing, since already इसमें दिखाया हुआ है कि यहाँ पे G1 phase and S phase में अलग अलग काम हो रहा है. ठीक है, it is also important guys. आपको ये याद रखना पड़ेगा because इसके रेगुलेशन भी वैसे ही होंगे. ओके अगर आपसे क्वेश्चन में ये पूछा जाए लाइक पार्ट बी पार्ट सी में ये पूछा लाइक अगर सपोज डीडीके इज गेटिंग म्यूटेटेड व्हाट विल हैपन टू योर रेप्लिकेशन. तो इट्स सपोज डीडीके इज म्यूटेटेड हाईली म्यूटेटेड यानी कि इसका हाई एक्सप्रेशन हो रहा है. ओके म्यूटेशन कैन बी हाई एक्सप्रेशन और नो एक्सप्रेशन राइट. तो अगर आपको बोला जाए डीडीके इज गिविंग अ हाई एक्सप्रेशन. ओके डीडीके आपके पास बहुत सारे कॉपी नंबर हो गए. व्हाट विल हैपन टू योर डीएनए रेप्लिकेशन? या completely mutate हो गया है. Okay, it is non-functional. Then what will happen to your DNA replication? Similarly with the SC, DKN, etc., etc. Okay, so these are the important proteins which I'm going to tell you. Rest all तो छोटे मोटे हैं वो हमें ध्यान रखने की जरूरत नहीं है. Okay, so important ones are first thing what happens? You have got the ARS. Onto ARS ORC comes, which is helping in recruitment of the MCM27. Okay, what is MCM27? It is mini chromosome maintenance. 2 to 7 proteins are there. Now this will help in the recruitment of DDK. Now what is DDK? It stands for your DBF4 dependent kinase. Sorry, dependent kinase. तो जहां पे भी K आ रहा है तो उसमें गैस मार लेना that it is your kinase only. ठीक है? Okay. So now DDK is helping in the recruitment of your CDC45 and SLD3. Now CDC45 again it is cyclic dependent cyclins 45 है. ठीक है? So they are the part of your cell cycle regulatory proteins. Now what is happening? The complex is being formed. Can you see? On to the MCM27, CDC45 and SLD is coming and bound to the helicase. So what exactly is happening? They are going to help in the helicase activation. ठीक है? तो वो कौन कर रहा है और SCDK. Now what is SCDK? ये आपका S phase का CDK हो गया. S phase CDK cyclin dependent kinase. S phase cyclin dependent kinase. Okay. Now it is going to activate all these proteins. Can you see all these proteins and helping in the recruitment of your Pol epsilon? Can you see it? Yeah. Okay, so you can see it over here. कुछ यहाँ पे भी हैं, कुछ यहाँ पे भी हैं, and your replication fork is getting ready. So Pol epsilon is getting ready over here. Okay, so this has activated your helicase. अभी helicase activate कैसे हुआ? See, you can see यहाँ पे आपका 6 hexameric unit था and यहाँ पे भी था. So ये उस तरफ चला गया, ये दूसरी तरफ चला गया. So this is your helicase activation. Now once this gets activated, your Pol delta, which is the main one, okay, and the alpha, which is the primase, all are getting recruited. Now they are helping in the, they are continuing the replication then. थोड़ा सा complicated है if you compare with the prokaryotes. Okay. Yes, guys. Any doubts over here? Like, please, I'll just remove the marking. Just go through the structure once. In your mind, you can just talk about it, and then don't worry, in detail we'll study again. ये तो कुछ भी नहीं है. This is just the starting of getting into the complex pathway of DNA replication in eukaryotes. अगर थोड़ा सा इतना समझ में आ जाएगा, then only we can go in the next complex level. Okay. So is this much clear to you today? Yes, Tanvi. See,
I'm not whole, but little.
It's okay. It's okay. And at the end में ऐसा होगा, you'll be like, अरे हमने पढ़ा ही क्या? See, prokaryotes में कैसे था? You were supposed to focus on the initiation, ठीक है, और termination. Simple था. Elongation के लिए कुछ इतना अलग से नहीं था. Whereas in the case of eukaryotes, initiation भी complex है, elongation भी complex है, and termination तो बहुत ज़्यादा complex है. Okay. आज के लिए अभी के लिए देखो, we are just going to start with the eukaryotic initiation and so many proteins are there. The problem is that you need to remember all the protein names and that in the sequence. ये थोड़ा सा problem है. And what I can suggest is go like a mathematic way. जैसे हम मैथ्स में इक्वेशन जो रहते प्रैक्टिस करते हैं ना, यू नीड टू प्रैक्टिस योर बायो के कॉन्सेप्ट्स, लाइक यू हैव टू ड्रॉ एंड सी. कि आप ऐसे ऐसे ड्रॉ कर सकते हो. ओके, वी हैव गॉट द डीएनए, यहाँ पे ARS है, इसके ऊपर ORC आया, ORC ने यहाँ पे MCM27 को रिक्रूट किया, देन DDK आया. ओके, along with the DDK you have got the CDC45 and SLD3. ओके, नेक्स्ट स्टेप क्या होगा? DDK is going to get bound to the MCM27. Then next SADK is coming. It is binding over to the helicase and separating the helicase. एक hexamer यहाँ जाएगा, एक hexamer this direction, right? We know the replication moves in the both direction, right? That is the activation, and for the activation you need these proteins. Then comes your delta and alpha for the priming and the replication. Okay. तो ये थोड़ा ड्रॉ करके या स्टेप वाइज लिख के यू कैन डू इट. Of course, nobody is asking you to make the beautiful structures, but you can just practice like that. Okay. Anything else, guys? I'm stopping here for today, and tomorrow we'll meet again. Okay. Any doubts are there in today's lecture?